This playlist collects together and organizes the individual component modules, which together will comprise our lecture on neurons and action potentials. The goal of this week's lectures, both this lecture and the introductory lecture to connectionist systems, is to provide you with a basic understanding of why it is and how it is that cognitive science researchers think of your brain as an information processing mechanism, as a computer. Now, many of us are probably aware that the average human brain has somewhere between 86 billion to 100 billion individual neurons. Now, each of those neurons, if we were to take an average across the entire brain, maintains about 7,000 synaptic connections, so connections with other neurons. This actually is a lower number than we have at certain points in development. So, by some estimates, gestating babies inside the womb grow about 250,000 new neurons a minute. Estimates for the number of synaptic connections for the brain of a 3-year-old are on the order of about 1 quadrillion, or 10 to the 15th power. Now, sadly, both the number of neurons and the number of synaptic connections between neurons declines as we mature into adulthood. It stabilizes for a while when we're in young adulthood, and then it declines further as we get older. Now, estimates vary for an average adult brain, but usually the figures that you hear are between 100 billion and 500 billion individual synapses. Now, what we want to understand in this lecture is how do those individual neurons work, and how do they communicate and process information with each other. Our goal then will be to understand the basic anatomy of neurons and the basic functionality of neurons. So we're going to start just by looking at the basic anatomy of a typical neuron. From there, we'll move to look at those components that are important in their overall functionality. We'll talk about the basic states of a neuron, and then we'll move into a discussion of action potentials, their basic function as cells. Finally, we'll end the lecture by talking about the different general types of neurons that are part of our central nervous system. Let's shift our focus then to the basic anatomical components of a typical neuron, starting at one end of the neuron and working our way to the other. We'll start by talking about dendritic branches, then the soma or cell body of the neuron, the axon hillock and the axon of the neuron, the presynaptic terminals, the end bulbs of that axon. We'll discuss glial and schwann cells, terminal fibers, and synapses, as well as neurotransmitters. A typical neuron is thought to have 4 morphologically or structurally distinct cellular components. The first cellular component we want to talk about is the cell body, or the soma. The cell body contains the nucleus, as well as other structures that are related to its metabolic functioning. The soma also functions to integrate the input from other neurons for the purposes of our action potential. The cell body has 2 different kinds of processes that project from it. On the one hand, we have dendrites, shown in the blue there. Dendrites are branch-like protrusions from the cell body which reach out to other neurons and facilitate communication. Dendrites are shorter than axons and taper as they extend from the cell body. Dendrites also exhibit multiple arborizations, or branches. The other vagination of the cell body is the axon, the extended protrusion from the cell body, which acts to carry electrical signals from the cell body to other neurons. It begins at the juncture of the cell body, and it has a particular anatomical feature that marks the beginning of the axon, called the axon hillock. And this is the region of the cell body where the axon emerges, and the axon hillock, as we'll see, plays a crucial role in acting like a gate to the axon for the purposes of our action potential. The axons themselves are oftentimes wrapped in glial or Schwann cells. Those Schwann cells tend to be in the peripheral nervous system and the oligodendrocytes in the central nervous system. These cells wrap themselves around the axon so as to create a sheath, which, as we'll see, facilitates the transmission of nerve impulses, covering parts of the axon in myelin, which is a fatty material. In mammalian brains, glial cells or neuroglia are the cells behind the neurons. Though they're not actually neurons, they're crucial to mental functioning. They provide the substrate that holds up the brain, and they help to maintain neural homeostasis by providing glycogen for energy, repairing damaged neurons, and clearing away dead neurons, as well as maintaining the ionic balance outside the neurons themselves. Glial cells sometimes even facilitate information processing functions of neurons. They form myelin, increasing signal speed, break down neurotransmitters in the synaptic cleft, and even release neurotransmitters and ATP. Axons, particularly in the peripheral nervous system, can be very long. They all, though, eventually terminate in terminal fibers or presynaptic terminals. These are branches from the axon ends which travel to other neurons and end with synapses on those other neurons. Of course, synapses are just points of communication between one neuron and another. The most common kind of synapse is a gap chemical-based synapse. A chemical synapse is a gap between one neuron and another, where one neuron will release that chemical, that neurotransmitter, and it will bond to the receptor sites of the other neurons on the other side of the synaptic cleft. Once these transmitters bond to the other cell, they affect the cell's state. And in fact, we should never forget that a typical neuron is loaded with neurotransmitters. And these include chemicals like serotonin that will act as signals when the neurons release them into the synaptic cleft in these synapses. Let's end this lecture module by briefly reviewing the structural components of our basic neuron. The central component of a neuron is the cell body, or soma, where all the cellular functioning structures are, like the nucleus and so on. Protruding from one part of the cell body are the dendritic branches. Those dendritic branches have postsynaptic terminals, sites where neurotransmitters can bind to the dendritic branches. The other side of our cell body here, we have the axon hillock, the point at which the axon protrudes from the cell body and goes along to the next neuron. Some, but not all, neurons in the human nervous system are myelinated. They are covered with myelin thanks to their cooperative relationship with glial cells or Schwann cells. Those cells then wrap themselves around the axon and act as a sort of insulation for the axon. The gaps in between those bits of myelination are called the nodes of Ranvier. And when the axon then reaches its destination, it breaks off into terminal fibers and ends in axon end bulbs or synaptic knobs.